everybody for joining in on the Spring Conservation Sciences seminar. Today we have Dr. Nathan Furey from the University of New Hampshire. And Dr. Furey is an assistant professor in the Department of Biological Sciences at UNH and heads the Fish and Movement Ecology Lab. He holds a PhD from the University of British Columbia, a master's degree from Texas A&M University, and a bachelor's from the University of New England. His research attempts to determine the motivation and consequences of animal movements through space and time, focusing on fishes of management and conservation concern. Nathan uses acoustic telemetry to track the movements of fish and crustaceans in estuaries and coastal waters to determine space use, habitat requirements, migration timing, and survival. Because animal movements require energy, the Fish and Movement Ecology Lab also conduct research in bioenergetics and predator-prey interactions. Knowing who eats who and quantifying diet quality allows us to predict consequences of ecosystem change due to warming waters, changes to habitat, and other anthropogenic impacts. So with that, I want to thank Dr. Furey for joining us today and turn it over to him.
Thank you, Chris, for the wonderful introduction. Thank you to the seminar committee for inviting me to speak with you all today. And I'm just really excited to have the opportunity. Before I begin, I do just want to acknowledge that where I am here and what is now known as Nottingham, New Hampshire, I'm speaking from Nadakana, which is the traditional ancestral homeland of the Abenaki, Pennacook, and Wabanaki peoples past and present. And I want to acknowledge and honor with gratitude the land and waterways that the Alnabak have stewarded throughout the generations. From a scientific standpoint, today's talk is going to cover several themes and several field sites and several projects. And so I'm here kind of as the mouthpiece for them, but they're only done through really the work of a lot of other folks. This includes students who aren't in my lab, such as Adam Canigan, who I co-supervise at the University of British Columbia, other professors really around the continent, a variety of state, provincial, and federal agencies, but also nonprofits and non-government agencies as well. And in particular, I want to thank and acknowledge the Honeygatine First Nation that gave us access to the first field site that we're going to focus on in today's talk. And certainly you're going to see a lot of these faces throughout the talk as well, but I need to acknowledge my incredible lab and incredible both graduate students and undergraduate students who help us do the research. And then lastly, we've been very fortunate to have a variety of funders and other supporters that help facilitate this work from a financial and logistical standpoint as well. So Chris gave a really nice introduction as to who I am and what motivates me, but I think this will allow you to understand the talk a little bit more as well. And so I am a fish biologist, but I also consider myself a movement ecologist. And this means I'm trying to understand what are the motivations and consequences of animal movements and what generates variability in animal movements. But if you study animal movements for long enough, you quickly realize that animals are moving either to feed or to keep themselves from being fed upon. So my lab also does a lot of work in trophic ecology or predator-prey interactions, trying to identify what factors influence predation or the ability, conversely, the ability to feed. And this action of feeding as well as movements, these are all actions that can either help you gain energy or lose it. And so my lab also does research in bioenergetics to try to quantify some of those consequences of different interactions. And then whether it's moving or feeding or using or conserving energy, these processes are all going to be influenced by where in the world you are. So we also try to keep in the back of our mind a landscape ecology kind of lens as well. So we understand how does spatial and temporal variability influence these processes. But again, kind of my passion and my bread and butter is in movement ecology. And so this led me to go to the University of British Columbia for my PhD and postdoc because I wanted to study animal migrants. Animal migrations occur across large scales where you have large contingents of populations all moving in a synchronous way across the landscape. And there might not be a more iconic animal migrant on the West Coast than the sockeye salmon that's distributed throughout temperate and subarctic watersheds. And these are diadromous fish, so they need to migrate from the ocean into freshwater to spawn. And they're iconic because they support these large commercial, recreational, and indigenous fisheries. They arrive to these rivers en masse, make the waters boil when they're abundant. We've all seen the nature videos of bears and eagles and wolves consuming these fish as they head upstream to spawn. Unfortunately, however, sockeye salmon are not doing very well. You can this is actually a smolt to adult survival ratio on the y-axis here, but you can just kind of consider it as survival. And we kind of see a decline through the 1990s and early 2000s. If we continue this out into the past 10 years, it's gotten even more bleak. This is showing a slightly different metric, just the yearly annual returns of these fish, and it's getting poorer. You might notice kind of the every 4 years, there's a spike. That's just kind of a characteristic of this stock in general, but we can see whether it's that 4-year period or all the other cohorts that we're seeing a pretty strong decline and pretty low numbers. This was exacerbated by a very large landslide that occurred in 2020. And when we think of salmon migrations, we tend to think of those adults because they're the ones that we see in the nature videos, and they're the ones that directly support those really important fisheries. But for the life cycle to be complete, there's another migration that occurs. And these are by juvenile sockeye salmon smolts. And this is when these juveniles that were rearing in freshwater head downstream and out to the ocean to grow nice and big before migrating back to spawn as adults. And there's been quite a bit of information to at least make empirical links between population productivity and kind of the success of smolts, potentially even more so than the success of those migrating adults. So my PhD and postdoc work occurred in this just incredible landscape here, Chilko Lake in British Columbia. And it's home to one of the largest sockeye salmon populations in British Columbia, and certainly outside of anywhere outside of Alaska. To kind of show you where that is on the map, it's that red arrow and location A. And so these juvenile sockeye salmon smolts have a long ways to swim. They swim through 3 river systems over several hundred kilometers before they actually enter into our estuarine or coastal environment of the Strait of Georgia. I mentioned that this is a really large population. I just wanted to kind of demonstrate that for you. We're just going to dip our head under the water here and take a look at these fish. And over about a 4 to 5 week period each spring, you have between 10 and 70 million of these juvenile salmon smolts leaving the lake and heading downstream. And my PhD really focused on kind of the behavior of these fish, how many of them survive downstream, what behaviors allowed them to maximize their survival. But we're going to take this in a little bit different of an angle today. In that this migration of these millions of fish that leave this large lake, but then get funneled out of the lake and into these relatively small, clear and slow moving rivers represents a resource pulse for consumers. So resource pulses are just simply when resources become highly abundant over a very short period of time, but often in a very predictable manner. So this is a resource pulse that occurs every spring, and consumers are likely to respond to it. And so we started a series of studies that focused on one consumer in particular, and this is the bull trout. So bull trout are a native char, so they're related to things such as lake trout or brook trout, and they're native to this system. And we just noticed that when we would arrive to this field site in early mid-April to prepare for the sockeye salmon smolt migration, that there'd be literally hundreds of these bull trout sitting at the lake outlet in the river, seemingly waiting for the smolt migration as well. So we started to do some diet studies to try to just understand, are they feeding on these sockeye salmon smolts? And If yes, how many? And the answers to that are simply yes and quite a few. So that image in the bottom right, that's the stomach contents of a single bull trout. There are 65 sockeye salmon smolts in that pan there. You can also see that they're in wonderful shape. If I had told you that that was just, some fish that we dip netted out of the river, you'd probably believe it. This implies that these fish were all ingested recently and over a very short time period. And in fact, when we dumped those fish into a cooler to process them further, some of them started to try to swim around again. I apologize. I know some folks are maybe eating their lunch while listening to this talk. So we did a lot of work characterizing the stomach contents of these bull trout and looking at their feeding rates and how it related to bull trout size and the really cold temperatures of the water that were being experienced. I'm not going to talk about that more today either, but we did have a few publications as well as some media through the Nature Conservancy that demonstrated that these fish are absolutely binge feeding. They're feeding at rates that greatly exceed what we think an ectotherm would feed at, given the cold temperatures. The water was often between 2 and 5 degrees Celsius when we were there. So I'm happy to answer questions about that and how we assess that moving forward. But the big take-home point here is that we have a resource pulse, and these bull trout are absolutely exploiting it by feeding at high rates. That leads us to kind of more of our more recent and ongoing research that I want to share with you today that's trying to ask the question, how important are these salmon resource pulses to bull trout? So it's very apparent that they're keying into them every spring and that they're feeding at high rates. But to what lengths are these bull trout willing to go to be able to exploit this resource pulse? And might it have any sort of consequences to fitness? And so to do this, I'm working with Adam Canigan, who's that PhD student at the University of British Columbia, who has done about a 3 to 4 year acoustic telemetry study on the movements of bull trout in response to these salmon resource pulses. For those of you who aren't familiar with this technology, it's simply the idea that we can take an electronic tag, we can surgically implant it into the bull trout. We can then place receivers or listening stations or hydrophones throughout the environment, and I'll show you where we placed those in a slide. But if the bull trout and its unique tags happens to swim by within range of that receiver and send out its signal, that receiver will know, okay, bull trout number 22 was just detected at 12:15 on Friday. So we get individual level information on the movements of these bull trout. We've done this for the sockeye salmon smolts as well. That's how we estimated their survival rates as they move downstream during my PhD work. But this shows you kind of the landscape that we're investigating. Where we have Chilko Lake, which is about a 65-kilometer lake in interior British Columbia. And the Chilko River is a little bit of a unique system in that it's a river that does flow south to north. And so those juvenile sockeye salmon stay in the lake through the winter, and then they're going to be migrating up the screen to the north out into the river to head downstream. All of those circles that you see there are those acoustic telemetry receivers, so we could track the movements of bull trout through the lake across multiple years. So this kind of area right here represents that lake outlet where we saw that binge feeding occurring. This is where the bull trout seem to like to hang out because it's a nice spatial constriction for those smolts to go through. So this is all still preliminary. Adam's just a couple of years into his PhD, but he's starting to write up his first chapter now. So I'm excited to be able to share some of these results with you. This is a little bit cherry picked because it's just 2 of about 100 of the fish that he's tagged, but he hasn't had time to update this animation for me. But these are the movements of 2 bull trout over a couple of year period. And you can see that, oh, there we go. You can see that time frame kind of going at the top there. And kind of the take home points here are that these fish come each spring, so come each May and April, they're back up at that constriction again to be able to feed on those sockeye salmon smolts. During other times of the year, they're using pretty much the entirety of that 65-kilometer lake, which implies that they're really willing to go a large distance or move a long ways to be able to exploit this resource pulse. And again, I'm only showing you 2 fish because that's what we have visualized, but it was roughly about 30 to 40% of the fish each year made movements that were consistent with this. And I'll show you some more data on this in a bit. The other thing that you might notice is that the fish often come back a bit earlier, particularly in the late fall, and not quite up all the way to that spatial constriction. We think this might be in response to sockeye salmon spawning. Closely related species such as Dolly Varden have been known to feed and even binge feed on sockeye salmon carcasses as well as on eggs when these fish spawn. So that's a nice little animation, but let's show this to you with some data kind of across the fish that were tagged. So now on the x-axis, we have time or month again, and then the y-axis is just simply of the percentage of fish tagged, how many of them are in that outlet, which is kind of this very small specific region that we have highlighted in red on the right-hand side. And so hopefully what you can see is that there is some periodicity here in that we're seeing in April and May, these fish coming back to the outlet. So these periods in red, this marks kind of the first and last date that we see sockeye salmon leaving that lake. This is counted through a government counting fence. If you think back to that video where we dipped our heads underwater and saw the masses of juvenile salmon smolts, that structure that was in the video is the counting fence. That allows us to have really high resolution information on when this migration occurs and the densities of fish that are migrating. It's certainly weaker, but we see potentially some smaller peaks occurring during these times that we have sockeye salmon spawning as well. However, we don't expect nearly as strong of a relationship with these specific receivers because there's a lot of in-river spawning. So we did see bull trout kind of blast through this area and actually continue downstream, but there's also a decent amount of lake spawning by these fish and by the sockeye salmon in the first 30, more like 20 kilometers on the northern side of the lake as well. So we see these fish returning and kind of aggregating themselves at this outlet, particularly for the sockeye salmon smolt migration. However, it's not all fish that do it. It's only about 30 to 40% of fish that do it. So this is just kind of a plot that again shows month by month, where are the fish throughout the lake in terms of distance from the outlet, between the fish that ended up in the outlet. So that's why in these yellow or orangish kind of color, they're very close to zero. They were found to be at the outlet at some point during that sockeye salmon smolt migration, versus the gray bars, these fish tended to stay further away. Although in 2017 and 2018, we did see those fish still aggregating or congregating towards the northern part of the lake. However, in the next couple of years, we see a little bit more separation. So there's certainly those fish that still come fall, even fall and winter, they're getting much closer to the outlet. But then there's another contingent of fish that are staying tens of kilometers away from the outlet and don't appear to be exploiting this resource pulse. So trying to tease out why would you move versus not, and what are the consequences of that, are kind of the next steps in Adam's research. And I'll just give you a little bit of a hint into that. But more broadly, this idea that bull trout are willing to move 50 or 60 plus kilometers each year just to be in this spatial constriction to exploit a migration was really neat and interesting to me, because I just kind of thought of this as cascading or coupled movements across landscapes where you have prey that are all migrating, presumably to exploit spatiotemporal variation in resources. And then that's actually enough to cue predators to move long distances themselves and potentially even migrating themselves as well. And so during my postdoc, I kind of reviewed the the types and strategies of movements by predators and consumers in response to migrant prey. And we coined this term of migratory coupling, where predators are indeed moving or are induced to move long distances in response to migratory prey. And we laid out hypotheses in terms of what types of systems and what types of taxa we are more likely to see migratory coupling or not. And that's kind of a topic that my lab is continuing to explore in new systems as well. But from a fitness or a condition standpoint, these fish should only be willing to move these large distances if it provides them benefits, because movements cost energy. Movements often mean that you're going into novel areas that you have less information on. You might have higher predation risk, etc. And so what Adam did, he didn't just simply tag these bull trout. Oh, and so certainly we used bioenergetics models to quantify that, yes, during this period, by feeding at really high rates, bull trout can maximize their consumption and growth. So they can grow at a much quicker rate than expected than if they weren't binge feeding. But then there's that kind of that potential cost side as well with the movements and such. So what Adam did is he didn't simply just tag these fish with acoustic telemetry transmitters. He also took fin rays from them, such that we had a non-lethal biopsy and a way to age these fish. So fin rays, although not often as reliable as otoliths, and then salvolinids, or the Char genus in particular, is notorious for being difficult to age. We were still able to develop some age and length keys for these fish, as well as age estimates for our tagged bull trout. And so he's able to develop kind of an age length relationship, such as this one, with all of the red dots being fish that had acoustic telemetry tags. He took a lot of other fish just simply for stomach contents. We were also investigating the exploitation of these fish as well via recreational and First Nation fisheries. So there was a lot of other fish that were handled and had biopsies. But if we just look at those red dots, we can see that we've got some relative fast growers versus slow growers, or red dots that fall above that best fit line, or ones that fall below that best fit line. And if we use those residuals as kind of an indication of condition, we can then compare the fish that seem to be experiencing migratory coupling by going up to that outlet during the time that they're tagged versus those that did not. And we actually see a pretty substantial increase here in the relative size at age for these fish that are experiencing migratory coupling. And certainly we then kind of assume or infer that if you're larger for your size, you're going to either one, have reduced risk, or two, have increased fecundity. So maybe some potential consequences here, but Adam's doing a lot of work with bioenergetics modeling, with sampling diets throughout the year for these fish to really try to understand the potential consequences of these complex movements, as well as just the variability in the population. And so just some brief lessons from kind of this first case study is that these resource pulses are going to provide feeding opportunities that consumers are going to try to exploit. And migratory coupling is one such strategy that these fish can use to maximize their feeding opportunities. But that unlike the sockeye salmon, where all of these fish are going to leave the lake and migrate, we see a much more complex and varied relationship here, that there's within population variability that we need to understand kind of both the mechanisms behind that, but also the potential consequences for it. So this is a system that's near and dear to my heart. It's where I did my PhD and my postdoc, and now I'm really blessed to be able to continue working in that system, at least vicariously through working with Adam so closely. But I want to also bring you a bit more in line with what my lab is doing with my own graduate students as well. So thinking about other systems where we have really strong resource pulses, there's one system that kind of jumps out, and I've been very fortunate to work in for the past few years. And that's the Arctic. Arctic coastal ecosystems are really just timed by resource pulses. They're driven by resource pulses. And this is because they transition from winters that are just pure darkness and cold for several straight months, and then transitioning to quite rapidly to a spring and into a summer that's completely light. So that's an image that's taken after 11 p.m. At our field site in Tremblay Sound and Nunavut, and it's just complete sunlight. This sunlight really drives a cascading series of effects on these coastal marine ecosystems. So that sunlight helps drive increases in temperature. That temperature is going to then affect the sea ice. And so this is just a quick kind of GIF or animation that you can kind of see how quickly this sound that is our study site that I'm going to mention in a second here goes from completely iced over to then iced off in July. That happens over a few week period. And so the temperature is driving that ice breakup and loss. This doesn't just allow the sunlight to penetrate the water and increase primary productivity. It actually creates a large pulse in secondary productivity immediately, because that ice actually has a bunch of little critters in it, whether it's algae and primary producers, but also a series of amphipods and other macroinvertebrates that live within that sea ice, but then as it breaks up and melts, gets released into the coastal marine systems. And so these systems are really well known for having this distinct kind of spring summer resource pulse, which then drives in a series of consumers into these systems. And so this is some work that a former graduate student of mine, Lars Hammer, and current graduate student, Nate Herman, are working on in close connection with one of my good friends and colleagues, Nigel Hussey, at the University of Windsor, a little bit closer to all of you than I am. And to give you a sense of where they were doing their work, the two dots here are where I am in New Hampshire, as well as the Chilko Lake in British Columbia. This is Tremblay Sound in Nunavut, Canada. So way up there. This is a remote field site. There's no lodging or anything like that. The closest infrastructure is in Pond Inlet. So the team, there's about 30 researchers that went up to this site yearly for about 5 years, and they lived in tents. But what's really neat is that even just a month before this photo was taken, the entire sound would have been covered in ice. So again, it experiences this really strong seasonality in ice cover. And when that ice comes off in the spring, you get a series of consumers that come in and exploit it. So this sound is really well known to the Inuit communities here, because every year, even though the sound is only like 5-6 kilometers across, you can see it in the photo, you can see shore to shore, you can have over 1,000 narwhal enter the system to feed. You can get just almost innumerable and kind of an incredible number of Arctic cod that come in, Greenland sharks as well. But my lab was there to study these bony fishes, and in particular, we looked at the movements and feeding of sculpin, which are resident to the system. They stay in this sound year round, even during that cold, unproductive winter, but then also migratory Arctic char. So this is another important Inuit fishery. Arctic char is generally the single largest contributor to protein of local communities in this area. And we wanted to know kind of what were the strategies between this migratory fish and this resident fish. Do they exploit the pulses? Do they exploit the pulses in the same way? And so the short answer here is yes, they're exploiting the pulses. So on the left is kind of a normal body shape for a sculpin. On the right, this is a sculpin that's just on amphipods. It is just feeding on as many of those ice-associated amphipods as possible. I'm not going to show you a lot of the diet data. I'm going to focus more on the movements, but the relative consumption rates of these sculpin were incredible. Generally, between 5 and 15% of their body weight was contained in their gut, which is like me having, 20 pounds of food in my stomach at once, which I certainly try to on certain holidays, but I do not get close to that. The char fed at relatively lower rates, more in that 3 to 6% range, but this is still quite high given their body size. But they are absolutely also exploiting this pulse. This is a stomach that's just chock full of amphipods again. So they're both feeding on it, but how are their behaviors or their movements influenced by this extreme seasonality in the Arctic? So Lars has a couple of papers out now demonstrating kind of the migratory movements of these char. And this animation will loop through. So if you don't catch it on the first time, don't worry, it'll come back around. But what he found is that for a specific subset of char that were actually overwintering in this kind of river system that you can't see on the map, but I'm labeling with a red arrow here. These fish would overwinter in the river and then actually come out into the sound and kind of extend themselves through more than half of the sound before that ice started to break up at all. So that's what you're seeing right now with the ice present. They're well spread out. And then boom, once that ice starts to melt, this means they're already well distributed throughout the system when that pulse starts. So kind of contrary to some of the literature, it doesn't appear that these char are just following the ice melt in a system. Rather, they're migrating in a way that they're already in position to exploit the ice melt as soon as it occurs. Just in case over Zoom, the animations aren't working very well for you, I'll show this in a static plot as well. On the y-axis, we have the number of our tagged Arctic char that are in the system or detected in the system. And hopefully you can see this really strong seasonality where during other times of the year when the ice is on, we just do not see char. The receivers are left in there year round. So if they were there, we would hear them. Instead, they come at really distinct pulses and they only stay for a month and a half or so before they take off. So this closely aligns with when the ice is there. So now the blue line is showing you kind of the ice coverage, and we can again see these fish start to enter the sound days before, or on average about a week before the ice comes off. And this was consistent between the 2 years. They then hang out for about a month, and then they're often leaving even before the ice is completely gone. So this implies that they're really there as that ice breaks up and those amphipods are falling out. So the char are obviously willing to move long distances to be able to exploit this resource pulse. But what are these smaller, more resident sculpin doing at the same time? So this is a nice series of videos here that Nate Herman has put together. This is from his master's work. He's getting ready to submit a couple papers on this very, very soon. But what you're going to see on the right-hand side is just that same animation of the ice cover. So you're going to see when it's an ice on versus ice off season. And then on the left-hand side, you're going to see the movements of the sculpin and how often they move among these gates. So each of these letters just represents a string of those hydrophones across the system. And we're going to watch how often do these fish bounce among gates. And it's going to start in kind of this summer or this ice off season. And so we can see quite a bit of movements of those fish early on in the summer, but it's a very small sample size. But then during the winter, the fish are not moving around a whole lot. But during the summer, once it comes back through again, we're going to see more of those intergate movements where they're bouncing among the letters there. And so this just kind of loops through a couple of times. I know it goes by really, really quickly, but if you even just focus on this box plot in the middle here, just simply the number of intergate movements per day is much higher for these fish in the summer than in the winter. So this implies that even these small resident fish are going to be bouncing around and elevating their activity levels more when the food is available during those cold, long, dark winters. To show this again in more of a static way, Nate calculated home range sizes for these fish during the ice-free and the ice-covered season. So this is just using kernel utilization distributions. And In the literature, generally, we tend to think of the 50% home range, which is these box plots on the bottom left here, as being kind of the core areas of use. On the far right-hand side, we have the 95% home range area. This is kind of considered the total home range in the literature. However, Nate just wanted to see, well, if we didn't really care about these kind of a priori defined home range sizes, do these relationships still hold? And they certainly do. So it doesn't matter if we're looking at 50% of the positions and using that to calculate a home range, or 95% of the positions to calculate a home range, or anything in between there. Very consistently, these sculpin are using more area during the ice-free season, during the ice-covered season. And we thought, even though this is intuitive, this was still interesting to us, just because, again, there's a lot of other predators and consumers that come into the system at these times. Things like larger Arctic cod that are large enough to feed on them. Arctic char would sometimes be seen feeding on sculpin, and then certainly narwhal, Greenland sharks, etc. So it must still be worth it to them to feed at those higher rates. And so kind of the take-home point here is just, this is a nice case study in demonstrating how, whether you're a migrating fish or a resident fish, you can still modulate your spatial behavior or your movements or activity levels to try to match and exploit resource pulses. So for the sculpin, they're likely just hanging out in an area, and then once they realize that there's food or whatnot, they're responding to it. While again, these Arctic char seem to be priming or timing their migration such that they're actually well primed to exploit the ice off rather than reacting to it. Okay, now I just want to take the last few minutes of my presentation here to bring us a little bit closer to my home and to the University of New Hampshire. I know it's not close to home for all of you, but where we're located in the Gulf of Maine within the western North Atlantic Ocean is a hotspot for climate change. We are seeing warming rates that are several-fold higher than our global oceans. We are in the top 1 percentile of warming rates in marine environments. And so we're trying to understand what are the potential consequences for that, for consumers, both for what feeding opportunities they have, as well as how it might affect their movements or space use. So I just want to give you kind of 2 projects that are ongoing. I don't have, any really profound results to share with you, but these students are doing such amazing work. In this case, Alia, who's an NSF GRFP recipient, that I really wanted to share with you. So seabirds, such as these common terns that Alia is working on, they undergo incredible migration. So they go all the way really across all latitudes of the planet throughout the year. But then when they breed in the summer, they are stuck to their breeding colony. So that breeding colonies, those islands are not going to move with climate change. And so we have about 20 years of data on kind of the diet of these terns, and where Alia is undergoing investigations to see how that's changed and how it relates to climate indices, how it relates to fisheries statistics on what types of fishes are available to them. But she's also interested in kind of the individual level behavior of these birds and how it might affect their foraging, what they might capture, and then what that might mean for the success of their chicks. So in that red box here, about 10 miles off of shore, off of New Hampshire's coast, is the Isles of Shoals, which is New Hampshire's only breeding colony of seabirds, including common terns and federally listed roseate terns. And she has used now for a couple of years GPS tags to track their movements. And so we're just going to see the 2021 movements of these birds playing here. And anytime that the color goes blue and it leaves then a little blue imprint on the image, these are locations that we have inferred that they are foraging. We use models that are called hidden Markov models to look at kind of the distribution of step lengths and turn angles of these animals. And anytime these animals make small tortuous movements, kind of like me, when I get to the ice cream section of the grocery store, that's when I'm going to slow down, really consider my options. That's when we're going to infer that these animals are foraging. So this allows us to identify these foraging hotspots of these birds, looking at individual variability. For example, a lot of these birds are going into our coastal estuary of Great Bay. Others are going down to the Merrimack River mouth. And then we've got a series of birds that feed offshore, as well as some that are going down to all the way to Cape Cod to forage. We can then link that with information provided by cameras and by people watching through blinds to see what types of fish are actually being brought back to the chicks. And then even taking it a step further, how well those chicks do. So we're looking at kind of these relationships between seabird and fisheries and forage productivity, both at these really fine scales. We're then actually going out to these hotspots and collecting water samples for eDNA and using modified purse seines and bait scoops to actually catch the fish in the hotspots to see what's available to them. But then also over these really broad time and space scales as well, through the incredible work by the tern biologists at the Isles of Shoals. For fish that are not tied to a single breeding ground, we're interested in how kind of responses to climate change and consequences to climate change are going to be species specific. So this is an NSF grant that we're in our second year of working on. And Nate Herman, who did the work on the Arctic sculpin for his master's, is now a PhD student in my lab working on this project. And it's the whole idea that here in the Gulf of Maine, and this image is showing you the distributions of National Marine Fisheries Service trawl surveys in the region that provide us estimates of distribution and abundance of fishes, but then also there's subsampling of diets as well. So we have literally about 10,000 fish diets that have been collected over the past 20, 30 years that we can analyze and integrate. And so, for example, some fish are going to move to climate change more than others. And the responses are going to be species specific, such that some are going to be likely to be more prevalent throughout the region. Others are going to be only more prevalent in certain waters. Other kind of more cold adapted fishes might likely get pushed out of the system. This has been documented by a lot of folks, such as Janet Nye, Kathy Mills, et cetera, that these distributions are shifting in response to climate change. We now want to know, what are the trophic consequences of that? How are potential predator-prey overlaps going to change? How have they already changed? And what are the potential energetic consequences? So this is some very, very preliminary data. Nate's presenting this as part of a poster at the Benthic Ecology meeting, very close to us in Portsmouth, New Hampshire next week. But he's essentially taken the diet of these 9 different important fish predators in the Northwest Atlantic, and then looking at shifts in certain feeding metrics across these species. He's also done it for each individual species. We just don't have time to go through all of that today. And what we're seeing is kind of the different regions. And so if we even just focus on one of these, the GOM, which is the Gulf of Maine, which is kind of our waters, I'll flip back to the map here. It's kind of the waters that are off our coast of New Hampshire here. We see that for metrics such as the percentage of fish that had an empty stomach, that's quite variable, but that's variable among fish just in nature in general. But kind of across regions through time, we're seeing an increase in kind of how general their diets are. And that's what that Levin's breadth index is a metric of. How specialized or how generalized are your prey types. So we're seeing a push towards generalist feeding. But even though they're eating a more generalized diet, the bottom row shows you that the relative consumption is decreasing. So this is how much is in the gut relative to the size of the animal. However, this might also just be an artifact of warmer temperatures and digestion rates increasing. You'd have higher turnover. So Nate has a lot more to investigate here, but this premise that diets do seem to be shifting in these areas across a wide variety of species that vary from specialists to generalists is really interesting, and one that he's going to continue further. So with that, congratulations, you've made it to my final slide. I know we've talked about a lot of topics today, but just some broad conclusions here. movements are a fundamental characteristic of life. And I really like to think of movements as that kind of that mechanistic link among our other fields of biology, you know, whether it's form or function, landscape ecology, predator-prey interactions, population dynamics. It's really movements that are helping link these, at least for mobile animals. How consumers are going to respond to resource variability is going to have consequences in terms of what risks they might experience, what types of physiological conditions they might experience, such as water temperature for ectotherms, and their feeding opportunities. And then we only focused on it in today's talk in regards to climate change and potential consequences, but humans are absolutely altering resource availability. I'm happy to talk about more examples of that in our lab during the question period, but this is going to have impacts on consumer movements and their energy budgets as well. So I hope you enjoyed this talk. That's all I have for you today. There's my email on the bottom. Always feel free to reach out to me if you have questions that you want to ask beyond this seminar. Thank you all again so much for inviting me today, and I hope you enjoyed the talk.
Awesome. Thank you, Dr. Furey. Just a reminder, anyone can unmute yourself or throw questions in the chat box. I will start off with a question for you though. So looking at or thinking about the bull trouts kind of congregating at inlets, do you have any kind of thoughts about which kind of specific fish are the ones that are congregating? Are they the older, larger ones, or?
Yeah, and that's we don't have a good sense of that right now, but that's something that Adam does want to investigate, and kind of at various spatial scales. So for example, something that I didn't show you today is that at the outlet for a couple of years, we actually used a high residency or high resolution positioning system. So you can throw a bunch of those receivers all really close together, and you can actually triangulate animal movements. And we see these bull trout on a daily basis going right up to that fence, right up to the narrowest constriction each night, and then heading back. But there's definitely variability in that and where those fish like to feed. And so we're almost curious, is there a little bit of a social hierarchy there? It does seem that some of the biggest and baddest bull trout really like to sit right up against that fence. And it might be because they're able to push other ones out. In terms of more broadly, which of these animals undergo migratory coupling versus not? That's a really, I think, important question, but one that we probably don't have enough data to kind of get at. I do think there's kind of an age component where as you get older, you're more likely to migrate large distances. And this has been shown in a lot of fishes, kind of the idea of partial migration or kind of ontogenetic shifts in migration. But we need to investigate it. It's also a little bit tricky because we think part of the reason why only 30 or 40% of these fish are undergoing this migration each year or undergoing it in consecutive years is because bull trout might also be going to other parts of the lake to spawn. And salvolinids are also really unique fish in that they don't necessarily spawn every year. They might only spawn every couple of years or every third year. So it could be they're mixing and matching these periods of feeding really heavily on fish versus, okay, let's go use that growth to undergo reproduction. So we don't have a good answer for you, but it's a really good question, Chris.
Great, thank you.
Other questions?
I have a question.
Yeah, go for it, Jackie.
Yeah, thanks for a really great and engaging talk. I was wondering, you kind of alluded to this, what are some other ways that humans are changing resources, and in particular with the salmon population decline?
Yeah, and so I can even, one of the things that we just didn't talk about at all today was that counting fence. And so what we found, so this is that fence that gets put into the river every year. And what we found is that bull trout that we caught right at the fence binge fed at a higher rate than the bull trout caught elsewhere through the system. So bull trout caught elsewhere in the system still binge fed. They are absolutely still exploiting resource pulse, but it seems exacerbated by this counting fence that gets installed, to the point that those bull trout fed at a rate similar to bull trout held in a lab that were just fed as many rainbow trout fingerlings until they literally wouldn't take food anymore. So what we call ad libitum feeding rates, as much as they can handle. So that fence likely just pushes all those fish in, and those bull trout can probably just feed as much as they want. And I have another graduate student, Tyler Parent, who's then looking at how do the juvenile sockeye salmon smolts respond to that. I can see if I've got that really quickly available. But in short, I don't know what my cursor is doing here, so I'm not going to. But in short, he's using the high resolution telemetry as well. And he's actually seeing these smolts go up to the fence, say, no way, I'm not doing that, and turn around and go back out to the lake. Sometimes for 4 or 5 straight nights before they then push through. And these are fish that are migrating, and they want to migrate really quickly. Once they're through that fence, they go 600 kilometers in a week. And they'll go 50 kilometers in that first night. So it seems to us that that fence is affecting the behavior of the prey, as well as then facilitating the feeding of the predators as well. So that's one example. Another example, again, I wish I could get my, oh, there, my cursor wanted to work well again. I also do work with some wonderful scientists in the Mediterranean who track the movements and population dynamics of dolphin populations. And then we use a series of kind of spatial models and such to link the movements of dolphins to different anthropogenic structures. And one example of this is that we found dolphins love trawlers, particularly really big trawlers that catch a lot of fish. So those numbers that you're seeing are the odds ratios of seeing a dolphin when one of those fishing vessels is present. So a small beam trawler, dolphins are only 4 times more likely, which is still a large effect, 4 times more likely to be seen if there's a beam trawler there versus not, versus these really large paired midwater otter trawlers, 30 times more likely to see a dolphin in the presence in one of those than not in the presence of one of those. So just some, I guess, varied examples as to how we're seeing how anthropogenic effects can influence animal movements. But great question, Jackie. Thank you.
Thank you.
Other questions? Maybe all the talk of consumption has folks either hungry or queasy. I'm not sure which. Just to show you how much those bull trout binge feed on those fish. Oh, excuse me. I don't know why it jumped slides on me. But just this will be my last photo, and then I'll let folks go. This is a stomach that's been dissected from a bull trout donated to us by the Honeygatine First Nation. And hopefully you can see it's just chock full of smolts. And if you can't see the smolts there, they're little eyeballs there for you to key them out. But yeah, thank you all so much. Again, for being here today. I really enjoyed it. Maybe sometime in the future, I'll make my way by the University of Minnesota and get to see some folks in person.
Yeah, thanks again, Dr. Furey, and awesome photos throughout. Just so you guys know, another seminar next week, same time. So feel free to join us, and happy Friday to everybody.
Take care, folks.